Hey everybody, we're just making the video for the homework, the group homework. So group homework 6, that supposedly is supposed to be a lot shorter math than the last one, but it's pretty much the same length. But anyways, the first one, we're talking about Niemann-Pick disease. It's a big D is non-diseased and little d is diseased. So the first one is A, what is the frequency of D allele in the population? To find that, basically, 1 over 150,000 equals either P squared or Q squared, whichever one you decide to assign it to. So you have to take 1 divided by 150,000 and take the square root, and that you get 0.00258. And then for B, to get the frequency of the D allele, you're just going to do 1 minus that, because we know P minus Q equals 1. So you're going to do 1 minus 0.00258, and that gets 0.99742. For C, it says, what is the frequency of the DD genotype? So for that, you would just take your P or Q, whichever one you decided to do. So basically the answer from B, and do multiply it by itself or square it. And that answer will be, it'll be 0.9974 times 0.9974, and that equals 0.9948. And then for D, for the heterozygous genotype, you do, instead of P squared or Q squared, you do 2PQ. So you do 2 times P, which was 0.9974, times 0.0258, and that you get 0.00514.
Okay. So number 2 was the fill in the blanks. Yeah. And for A, it asks, what is the term in the Hardy-Weinberg equation that represents the frequency of the IB allele? And IB in blood type, there's IA and then IB. So the IB is the second one, so it'd be Q. While the term in the Hardy-Weinberg equation that represents the frequency of the IA and IB genotype, so we have the P and the Q together. So to find that, we do 2PQ. And then for B, it says the DiGeorge syndrome is an autosomal dominant disorder that affects 1 in 4,000 individuals in the human population. The 1 in 4,000 individuals in the population affected by DiGeorge syndrome is represented by the P term in the Hardy-Weinberg equation. It's P squared because it's the autosomal dominant disorder. And so since it's dominant, that's going to be the P. And so it's P squared. C, this question refers to the situation presented in part B, the one before. The 3,999 in the 4,000 individuals is the population unaffected by DiGeorge syndrome, and that is going to be represented by the Q squared terms in the Hardy-Weinberg, because that's the recessive trait, so that's the Q. And then D, the type of plumage found in mallard ducks is determined by 3 alleles at a single locus. M subscript R produces restricted plumage, M produces mallard plumage, and M subscript D produces dusky plumage. And then it goes on to say the restricted phenotype is dominant over mallard and dusky. Mallard is the dominant over dusky. So the M subscript R is dominant to M, which is dominant to the M subscript D. And so it says the blank term in the Hardy-Weinberg equation best represents the frequency of the MD allele. So that was the least recessive. And so when we have 3, there's going to be the P, Q, and the R. And so the R represents the M subscript D allele because that is the most recessive. It's the least dominant to the other 2. For E, it says the question refers to this situation presented in D. So that question before with the ducks. It says the blank terms in the Hardy-Weinberg equation represent the genotype of a duck with just restricted plumage. And so that's the most dominant. So that can be 2 M subscript R's. It can be an M subscript R and an M subscript, and an M. And then it could be an M subscript R with an MD. And so the P squared gives us the MR, 2 of those. The 2PQ is the MR with the M. And the 2PR is the MR, the MR and the MD. And so those are the 3 different ways that we can get restricted plumage. And the human disease sickle cell anemia is the result of an SNP. For G, monomorphic is what it's called when there's a trait that produces a single phenotype in 99% or more of the population. It's not the polymorphic. And H is DEAMS. That's another name for local populations. And the MHC gene is the gene in humans that is highly polymorphic.
Sweet. So in 3, it talks about Marfan syndrome, and it's an autosomal dominant disorder. For the purposes of this problem, we assume that 1 in 6,000 people have Marfan's. So basically, if we know it's dominant, we can associate it with an M letter. So big M would be, you'd have Marfan's, or big M, little m, you'd have Marfan's as well. So if A is asking for the frequency of the non-diseased allele in the human population, it's asking for everyone that's little m. So we could say it's Q. So Q squared would equal the genotype population. So if we want to get the phenotype of just a single allele, we have to do the square root of 599 or 5,999 divided by 6,000, because those are the people that are unaffected compared to the 1 in 6,000 that is affected. So when you do that, you get 0.99992. And that's how many people, or 99.992% of people are unaffected. So then to get the frequency of the people that are affected, or the frequency of the M, the dominant M allele, you would do 1 minus Q. So we did 1 minus 0.99992. Then we got 8 times 10 to the negative 5th. We know that was a little off to the study or the answer key, but we rounded and we just used the previous rounded instead of the actual square root. And then to figure out the heterozygous, you do the 2pq. So now that we have our p and we have our q, we do 2 times the answer to A times the answer to B, and that would get 1.599 times 10 to the 4th. And then so if we know that's the frequency of people that have the heterozygous allele, when it asks if there's a population of 5 million, you just take that frequency times it by 5 million, and we got 800 people. I know the study, or the answer key said 833, and that was just due to the rounding error in B. And then for 4, this one's got a lot of math, a little more complicated. But basically, we need to figure out the frequencies for the R squared and the R to third alleles. Basically, that means you want to figure out how many of those alleles are in the population, not the 2 together, but the actual separate. So for A, you do 2 times 135, because in the first genotype, the R squared and R squared, there's 2 of those alleles, so you have to multiply by 2, plus 44 because there's 1 R squared in that, and then you divide it by the total number of alleles, so 380. You get 0.82632, and that's the P. And then you could do 1 minus that answer to get the second one, or you could do 2 times 11, because there's 2R cubed in the 11 number of 11 population, plus 44, because there's 44 R3s in the second, and then divided by 380, and you get that as well. And then B is a little bit more difficult, so kind of hard. Basically, you have to figure out the expected genotype for each one, for the P squared, the Q squared, and 2pq. So how you do that is you take what you got for your P, multiply it by itself, or square it, and then times it by the total number of people, or total number of, we're doing it for a locus, a pine tree. And you get the expected of 1 to 129.30 or 73. You do the same thing for Q. You take what we got for Q in part A, which is 0.1737. Take the square of it, times by 190, and you get 5.73. You do the same thing for the heterozygous, which is 2pq. So you do 2 times P times Q times 190, so you get 54.5. And then what you'll do is you'll do the chi-squared test for every single one, for P squared, Q squared, and 2pq. And the chi-squared test is the sum of all of those added together, and it's observed minus expected. And you take the square root, or you square that, and then you divide it by the expected as well. So for example, for the first one would be 135 minus 129.73 squared divided by 129.373. and you get 0.21474. And then you do that for each one of them, and you get your x squared value, and your x squared value equals about 7.1. And then with that, what you have to do is figure out your degrees of freedom, which is the number of categories minus 1. So we had 3 categories. You minus it by 1, it's 2. Compare it to the chart that we have, and then figure out if we reject or support the null hypothesis, or not reject the null hypothesis.
Fail to reject.
Yeah, fail to reject is the correct word. And this we reject it because the chart, we were too high. And it could be multiple reasons. It could be because the population size is too small. Could be having our third gene is harmful, so they die off. All sorts of different possibilities that could happen.
Okay. All right, so 5, it talks about 4 different kinds of dogs, or different alleles. And in this order, they are dominant to the others. So the number 1 is the chinchilla dog, which is CCH. Next is the white dog with dark eyes, which is CD. Next one is the pale gray dog, which is CB. And the next one is the albino dog, which is just C. And so it says that we assume that the particular population of these dogs is in Hardy-Weinberg equilibrium. The allele frequencies for our 4 alleles are. And so it gives us the different frequencies for the alleles. So for A, it asks, what is the frequency of a CCH, a CCH? And so for that one, we take the CCH, the frequency is 0.37, and we're going to square that to get that number. And that number squared is 0.1369. The next one is the CB and C alleles. And so CB, the frequency is 0.25, and for C, it's 0.15. So we do 2 times 0.25 and times 0.15. And for that number, we get 0.075. And then for the last one, asked what is the CC genotype. And so we take that frequency, which is 0.15, and we square that number, and we get 0.0225. The next one asks, among 2,000 dogs, how many would you expect to have the pale gray phenotype yet be heterozygous? So pale gray is it's dominant over only the albino dog. And so if it has to be heterozygous, it's going to be CB and C for the genotype. And so for that, we do 2 times the frequency of CB, which is 0.25, times the frequency of C, which is 0.15. And we get 0.75. And we times that by the number of dogs, which is 2,000. So we get 150 dogs for that one. And for C, let me just need to see that question. So it says, among a population of 5,000 dogs, how many would you expect to have a white coat with dark eyes phenotype yet be heterozygous? So this one is dominant to the pale gray and the albino. So there's 2 different ways that it could be heterozygous here. So it's CD, which is its allele, CD and CB could be one form. Another one is CD and C. So we're going to take both of those together, and we're going to go 2 times the frequency of CD times the frequency of CB, which gives us 0.115. And then we're going to take 2 times the frequency of CB times the frequency of C. And that's going to give us 0.069. And so we take both of those different frequencies and multiply them by 5,000, which is the population. And one gives us 345 dogs. The other gives us 575 dogs. And together that gives us 920 total dogs, which would have that phenotype of the white coat with dark eyes, but it's heterozygous. And the last one for D, it asks, among a population of 15,000 dogs, how many would you expect to have the CCH, CB, or the CC genotypes? And so what we do here is we take the CCH times the CB, which is 2 times the frequency of CCH, which is 0.37, times the frequency of CB, which is 0.25, and we get 0.185. And we multiply that by 15,000, and that gives us 2,000. 2,775 dogs. And then for the next one, we take the CC value, and that's 0.15, and we're going to square that because it's just with itself. And that gives us 0.225. And for the frequency, so we multiply that by 15,000, which is the number of dogs. And that gives us 337.5 dogs. So we take those two numbers and add them together, and we get a total of 3,113 dogs total for it. And that's what we got for this one.